Hello everyone, my name is Rafael. I'm a fifth year PhD student in electrical engineering at NC State University. And today I'm going to present the work that we published in IEEE Sensors 2020. Let me share it real quick. Which is a feasibility study of water stress detection in plants using a high-throughput, low-cost system. This work has been done thanks to the United States Department of Agriculture, the Assist Center, and this list of authors, which I am the first. So let's start talking about this project, starting with the motivation involved with it. So the motivation lies on water deficit in crop management. Ideal water management enables optimal crop development, implying high yield and canopy. Identify water stress early or before it is visually apparent may allow measures to reduce such impact. Specifically maize plants, which is our object here, the youngest expanding leaf, which is being represented here with this green box, which we'll be calling the yield, has been shown to be sensitive to water stress, as you guys can check in the references mentioned at the bottom. So our purpose here is to try to use or try to do visual plant phenotyping and use that as a useful technique to identify the stress condition. And in visual plant phenotyping, we're basically trying to infer the physical properties of the plant based on the images that we take from them. So let's talk about the contribution that we left with this proposed work. We developed a system capable of acquiring a high volume of images, which is being represented here on the left, on the right, sorry, when we were testing it in our office. Those part of the state-of-the-art rely on IR measurements or sophisticated hyperspectral or thermal sensors and cameras. You can see a brief list of the state-of-the-art papers and more can be checked in our paper. This system is intended for ground level measurements as opposite for IR measurements and using more accessible equipment. We want to make a proof of concept with this paper for identifying early stress with low cost equipment. So now that we presented our system, let's talk about in more details about this high throughput data collection system. The system represented on letter A, which is the same one represented in the last slide, relies on a near-infrared camera covered with a blue light film filter. This filter blocks most of the red and the red and the green light spectrum and allows most of the blue and the near-infrared wavelengths to come into the camera. Oops. So everything is controlled with a microcomputer, the Raspberry Pi, which is represented with this gray block here. The Raspberry Pi controls all the actuators that are doing the movements here in this data collection system. The plants are scanned by repetitive movements, intended for automating some steps of image processing. Like when we want to block some undesired parts of the images, we take advantage of the repetitive movements of the platform for data collection, because with that, we know where exactly the picture was taken. One full scan of this prototype takes about 40 minutes to finish. And here we have a card that travels in this vertical degree of freedom, which is sliding the camera. And we also have another motor driving this horizontal degree of freedom to do the vertical scans with our platform for data collection. So we use two plants, one for water stress and another one for being well watered. And the species are specifically the Syngenta Egregia Viptera. They're represented here at letter C, where we placed them already in the chamber that we used. And in this chamber, we were able to control temperature, humidity, and also lighting. And we can see at this figure on the right that our platform for data collection was already placed there. However, we had to use extra NIR lighting with some LED illuminators, as we can see in this figure D, because as opposite to the lighting, the sunlight that we had in our office, in the chamber, we needed to have these extra LEDs to have enough FNIR. So let's talk about the method that we use to use that data collection system for our visual phenotyping approach. So the NGB images that we obtained with the camera, the red channel was actually replaced with a near infrared channel, so NGB. So this image is converted to a high value saturation color image, which is represented at letter C. And we can see the steps from the original image on letter A, the NIR image on letter B, and the HSV saturation in C. A binary mask is then created based on a threshold, and then we apply this binary mask to the original NIR image, and we obtain what is being seen here in this mask NIR. But you can see that the mask NIR doesn't tell much about the reflectance of the leaves. That's why we attribute these colors that are represented here on letter F, that we call the pseudo-color scheme diagram, where the brightest pixels are represented more in orange color, they're not as bright as yellow, and the lowest brightness pixels in green. For extracting the yield, we had to use a detection algorithm that we relied on a deep learning model called Detectrum. The reference is also listed here. And that helped us a lot to extract the yield and then compute the information specifically to that leaf. And we can see that with this technique, we're able to compare the drought stress plant here at the top with the well-watered plant at the bottom. And we can see as the experiment ran, and it took like 11 days, that we are able to see the difference in reflectance from the two groups. So you can see that the ones at the bottom reflect much more light than the ones at the top, because they're much more red than the ones at the top. And with that, a histogram of the segmented leaves using the pixel values are then obtained. So let's show now the interesting results that we obtained, along with some experiment and discussion details. So for each plant, the well-watered represented here at the bottom, and the drought stress at the top. The pixel values were normalized because that was a necessary step to construct the histograms, and we obtained them. We compared the distribution values over time with the first day of experiment for each plant. So the first day was our reference, and we compared how the distribution changes over the other days compared to the first one. And we can see here that on the top, we have the well-watered plant, but on the left, we're representing all the leaves, the pixel values for all the leaves. And on the right, only the ones related to the yield. And at the bottom, the two at the bottom are related to the drought stress plant. So we can see that the well-watered plant, in terms of yield, the distribution pretty much shifts to the right over time, while the one that is under the water stress pretty much doesn't change, it doesn't move in terms of reflection, what makes sense. And another thing that is interesting when we look for all the leaves, for the well-watered plant, we have the distribution skews to the right over time, and the one that is being stressed is skews to the left. So that's very interesting results. The comparison was done by using a divergence metric for probability distributions, the Earth Movers Distance, EMD. The EMD is a metric that essentially quantifies the amount of effort to transform one distribution into the other. We also tried the Kullback-Leibler divergence, the Shannon-Jensen divergence, but they didn't work as well as this one. So that's why we stick to this one. So a little bit of more results. By observing the distribution of all the leaves, which is being represented here on the left, the red line represents the well-watered group, and the blue one represents the drought stressed. We can see that after day 4, the difference between the two become drastic. And it's important to notice that at day 5 is when the water stress actually became apparent for the human eye on the leaves of the plants. And when we compare the pixel values only for the yield, the difference is even more drastic. The same thing is corroborated when we compare one plant to the other, which we're calling here a cross-check comparison, where we're comparing the well-water plant to the drought-stressed one. If we compare all the leaves on the left, we see that the difference comparing to the yield is much different. So the yield is much more telling than all the leaves together. This shows that there is potential for identifying water stress before it becomes apparent. So with that, we're able to talk about possible avenues for future work and some final thoughts we had for this study. The first one is based on an observation that we did that data annotation for yield turned out to be very time consuming. Active learning or multi-instance learning could be used for reducing labeling efforts. In active learning, we have pretty much a oracle that helps the algorithm to attribute labels to the samples. And from multi-instance learning, we can use a course level detail label to find fine level details in the data, for example. So let's say if we label the leaves with bounding boxes, the algorithm is able to find the contours of the leaves from that. So that reduces a lot the annotation effort. So with that, we believe that predictive models present a potential to aid automatic detection of early water stress. And further data collection is indeed needed with more plants in order to prove repeatability and robustness of obtained results. With that, I thank you for watching my presentation. I hope to hear your questions soon. Bye.